Hello, we are going to be continuing our notes on the cell parts and functions. We're going to start off by talking about the cytoskeleton. This is found throughout the cell. It could be tubes, it could be strands, it could be different filaments. Pretty much they're made of proteins, and they're going to give the cell its shape, internal organization, movement of the actual cell and their organelles. It provides the framework of the cell. It's kind of like scaffolding that's going to help provide the structural support, the cell shape, and so forth. Okay, there are three types of cytoskeleton that we'll be talking about. This is just showing you an example of what the cytoskeleton can look like within the cell. Again, like scaffolding. Microfilaments are more solid and thread-like, and they are made from the protein called actin. This is what's going to provide the structural support and assist in the cell moving and changing shape. So microfilaments are a little bit more thread-like. Microtubules kind of look like ziti, and they are made up of the protein tubulin, so they are more hollow and flexible. This is the scaffolding that supports the cell, and it also creates pathways for the organelles to move throughout the cell. Intermediate filaments are more rope-like in structure, and they're made more of a protein that is more fibrous. And that is going to help to reinforce the cell shape and to hold the organelles in place. So you can see here we have the microfilament, which is going to be very thin thread-like, the microtubules, which is a little bit more hollow, and the the intermediate filament is more rope-like. We also have cilia and flagella. Pretty much, microtubules are wrapped around an extension of the cell membrane. They are going to allow the cell to move. Cilia are much smaller. They are short, hair-like organelles, and they typically occur in larger numbers and are densely packed together. So they will kind of act to propel the organism through water or such, and they kind of act like a rowing. You have to go in the same direction. Where the flagella is more larger, and it's going to be more undulating motion, and it's typically much longer. Showing you the cilia and then the flagella. Again, both are used for movement. Okay. Centrioles are going to be very important in animal cell reproduction. So this is only found in the animal cell. And we do find this above the nucleus, and to me it looks like a stack of firewood. And it is made up of microtubules. So we could kind of call this like the reproduction center. And we'll talk more about this as we talk about the cell cycle. Okay, the centrioles will move to opposite ends of the cell as the cell starts to divide. Okay, so now we're going to talk about the cell membrane or the plasma membrane. This is found outside of the cytoplasm, and it's going to surround the cell. If you remember when we talked about the phospholipids and the lipid part of organic chem, we said it makes up the cell membrane. So we call it a phospholipid bilayer. Okay, so the main function is that it determines what can enter and leave the cell. And this is just showing you what that lipid bilayer would look like. So remember, you have your phosphate group, your glycerol, your fatty acid tails. Very fluid. This can move, and things can enter and leave the cell. Okay, just again showing you we have a hydrophilic end, and we have the hydrophobic end that does not like water. Okay, so it is selectively permeable, and we'll talk about this when we talk about the cell transports, but we are going to determine what can enter and leave the cell. It's just showing you the barrier or the outside edge or the cell membrane of a red blood cell. Again, just showing you outside barrier. Now, both plants, animals have this, bacteria has this, fungus, bacteria, and so forth. Okay. I just love pictures here. I'm just showing you all the different parts of the cell membrane. Okay, Now, cell wall is only found in plants, bacteria, and fungus. Okay, it does not determine what can enter and leave. It is going to provide support and protection. So it's an outside of the cell membrane. Okay, and it is going to be very stiff. All right, it does not determine what can enter or leave, and it does remain intact once the cell dies, so it does not disintegrate. And that's what Robert Hooke looked at when he was looking at the cork cells. He was looking at the cell wall. Okay, now there is some channels that are going to help with plant cells. Okay, the plasmodesmata, okay, it's a channels between adjacent plant cells that is going to allow for communication and movement of things between the two cells. Okay, okay. This we talked about when we talked about the nucleus, but I wanted to just have you write this down. The nuclear membrane is a nuclear envelope. Again, it is surrounding the nucleus with a double membrane, does have pores, and things can move in and out of the nucleus. Okay. Again, this was the plasmodesmata that we talked about with the plant cells, allowing things to communicate and move within and out of the plant cells. Okay. Animals are going to secrete a sticky substance, which is called the extracellular matrix. That's going to help hold cells together and help protect and support the structure. Okay, we also have junctions, just like the plant cells had the plasmodesmata. We have tight junctions, anchoring junctions, and gap junctions. Okay, tight junctions are found within the digestive system. Very, very tight, kind of almost leak-proof. Where the anchoring is going to be found in the skin and the heart, and it's going to form a strong sheet. Okay, and the gap junctions, well, there's gaps in it, so small particles can flow through, and we're going to see that in the muscular system. And just to show you the different pictures of what they are. If you have any questions, please see me tomorrow. Thank you.